student did Hershey's and Chase experiment. So what happened in Griffiths? What he proved?
That something is
something is transferring from one organism to another organism. And interestingly, even then, so there is a smooth strain and rough strain. Even after giving smooth strain, still it can able to transfer this to the rough, making the conversion of rough to smooth, inducing virulence in the host. That is all about Griffiths. But he don't know what is transfer. But he proved that these things are inherited or transferring. So it is called as transferring transfer. Then what everything? Now everything you know what is the transferring molecule? Is it a protein? Is it a lipid? Is it a DNA? So for this purpose, what he did? So he tried to inject all these things into the same rat, and he concluded that it is the DNA that is responsible for the gene transfer. That is killing. Yeah. So that is all about the Hershey's experiment. And after find this Hershey's Chase experiment. What they did in Hershey's Chase experiment? So he used a bacteriophage. So in bacteriophage, he used two types of isotopes. One sulfur 35, and this is P32. So separately. So first he did with the coating. He introduced it to the bacteria. Then he centrifuged it, and he started checking the things. And when he used protein, it is insufflated but not in the bacteria, so it's not protein. When he did the same test with P32, that is genome, here we got we detected in the bottom, which indicates that DNA is the responsible molecule for gene transfer. So that is all about these three experiments. So these three experiments given or paved the way to the next generation scientists up to the 1970s only. 1952, 1960s, that's it. So this is a whole very old research. So they try to understand, and the godfather for all these things is who is the one? Who is the father of genetics? Yeah, Gregor Mendel. Gregor Mendel is considered Mendel as considered as the father of genetics. So he did experiments on pea plants, and he concluded there is something that is transferred to the next generation. But you don't know what is it. That is it. Next. So let me see your syllabus. So DNA as a genetic material we understood. Flow of genetic information, structure and replication of gene. So flow of genetic information is this. These three experiments concluded that genetic information can be passed from one generation to another generation. That is flow of this one. And in the morning we also discussed about central dogma of life. So what is the central dogma of life? What is the basic concept? We have a DNA converted to RNA, and RNA converted to protein. Right? So conversion of DNA to RNA is called as transcription. This is called as translation. So let us go into that. So DNA is stored in the nucleus. Right? Now the DNA will be unbinded. The DNA will be unbinded wherever the portion we need to unbind. Here it is unbinded. This portion is unbinded. So what is the purpose of having DNA? It has the information, right? Say for example now, assume this is a cell, and now the cell need to code a protein, maybe hemoglobin, right? Let's say. So out of this entire genome, wherever the information present regarding hemoglobin, that particular part of gene will be DNA will be unbinded. After unbinding, a piece of complementary strand will be provided, will be generated. That this is called as, this piece of strand will be coming outside. That RNA is called as messenger RNA. This RNA has the message to code a hemoglobin protein. That is called as messenger RNA. Now this messenger RNA will exit the nucleus. It will exit the nucleus, coming outside the nucleus. Once it is coming outside the nucleus, now it don't know what to do because there is a very big cytoplasm, very big group, right? Now, outside there is something called ribosomes. Ribosomes. It is the ribosomes that are responsible for making proteins. Now, someone should help the RNA to reach the ribosome. For that purpose, we have a small, this is mRNA. Now, there is something called tRNA. This transfer RNA will take it to the ribosomes. Okay, tRNA. Now, in the presence of tRNA, it will be given to the ribosome. By the way, ribosome is just an RNA. By the way, ribosome is made up of ribosomal RNA. RNA, if you know. RNA. Okay. So now, this ribosomal RNA will start complementing this, and they will start doing something called translation. Do you know how translation works? So that we need to understand. In order to understand translation, we need to understand the detailed structure of the DNA. So far, I will tell a few more stories now. So what happened was, the scientists understood DNA plays a vital role. Now they need to know what exactly the DNA is. What this DNA is made up of. No one knows. They concluded that nucleus has some DNA. Some nucleic acids are present. Why? What type of nucleic acids? These acids has a ribose sugar. So they have sugar and some acid is there. So they thought ribonucleic acids. Ribose sugar containing acids which are present in the nucleus of a cell. Hence they are called as ribonucleic acids. But they don't know what are those ribonucleic acids. Okay? And also when they check the oxidative and reductive testings, whether it is uptaking oxygen or it is removing oxygen, they came to know that they are deoxygenated ribonucleic acids. They are deoxyribonucleic acids. That is what they understood. Then they started classifying the ribonucleic acids. Once they started classifying, they came to know that in the nucleus, they identified 5 types of molecules: adenosine, tyrosine, guanosine, cytosine, and also in RNA, in ribosomes, they also identified one, that is uracil. So they came to know that there are 5 types of ribonucleic acids. Deoxyribonucleic acids or ribonucleic acids. Okay? Yeah. If it deoxygenated DNA, if it is not deoxygenated, then it will be RNA, ribonucleic acid. Okay? Fine. So now another puzzle. How to crack this story? What is the structure of DNA? Now they came to know what DNA is made of. They came to know it made of some bricks. Now they want to know the arrangement of those bricks. How these 4 are arranged. How is it going on? They didn't understand exactly. So they were trying to rule out the reasons, and they came to know the fundamental ingredients of these sugars are commonly they have a ribose ring. They have OH molecules. Okay, if OH is not there, oxygenated, then it will be oxygenated, deoxygenated, and also they found phosphate. Phosphate is there in the nucleus. So there is a presence of phosphate, there is a presence of ribose sugars, and there is these 5 base pairs, these 5 nucleic acids. I can't even say base pairs, so future they discover. Then, when they are working on this one person came. His name is Chargaff. C-H-A-R-G-A-F-F. Chargaff. He did one wonderful experiment. What is that experiment? He started measuring he took 1 ml of sample. He took 1 ml of sample, assume, or 1 gram to make things easy. 1 gram of sample. What he observed was, if adenosine is 50 milligrams, then tyrosine also 50 milligrams. If guanosine is 20 milligrams,
Then cytosine is 20 milligrams. There is some pattern among those molecule proportions. If A is number of A is equal to number of T, number of G is equal to number of C. But no one knows why it is like this. So he came up with that. Definitely A is always binding with T and G is always binding with C. But you don't know how exactly they are binding. Then he started further molecular bonding structures. When he started understanding what type of interaction they are having, in between A and T it has a triple bond. In between G and C it has a double bond. So now we are getting some more story, details in this story, in this puzzle of understanding structure of DNA. Clear? So definitely A binding with T, G binding with C. So we are getting even more clear picture. Then how these are interconnected, how they are structurized, how they are framed to work each other. And there are certain rules we have already understood. They are so strong that they won't lose their rigidity, they are maintaining it, and also they are passing it to the next generation, and they are also recreating themselves. How is it possible? We are getting the story, but we are able to comprehend how these things are made up of. Then, you don't know one story, I didn't say it here. Anyway, I will let you know later. There is a wonderful woman that the textbooks won't discuss. I have to discuss that. Fine. So let me give you some basic arrangement of these things together. So first, there is this phosphate. Accurate diagram. I hope I can show that. So GC, I am resolving GC has triple bond and AT has double bond. So okay, AT has double bond whereas GC has triple bond. I said it was clear. Then they said like this. The basic structure of a nucleoside is like this. It has a phosphate backbone. To the phosphate backbone there is a sugar molecule. To this sugar molecule there is a base pair, nitrogen containing base pair. A phosphate connected with a sugar molecule. This is connected with a base pair. This is the basic structure of a DNA. This is called as nucleotide. What is the difference between nucleotide and nucleoside? When you are seeing the whole structure along with the phosphate, sugar, base and this one, this entire hyperstructure is called as nucleotide. TIDE, nucleotide. Okay? I can do a beautiful diagram. Let me check where is it. I don't want this graphics. Let me. See, a phosphate, sugar with a base pair together called as nucleotide. If you only see a sugar and molecule, this is called nucleoside. Sugar and the base pair is called nucleoside. If you connect it with a phosphate, then it becomes a nucleotide. Side will be inside one, tide means together. Phosphate, sugar and base pair together is called as nucleotide, and this is called as nucleoside. Actually, there is one simple diagram where we can clearly differentiate these two. So, see, this is how the DNA was structurized. We have a phosphate backbone on both the sides. Phosphate backbone. To this phosphate, there is a sugar link. To the sugar, each sugar, one base pair has been attached.
If it is T, always attach with A by double bond. If it is G, always binding with C by a triple bond. Okay? Keep in mind, we still don't know this organization. We came to know later. Previously we don't know. We are already understanding the basics. So far we understood that there is a base phosphate, then sugar, and a base pair. A or T or J or C. Okay? Then all the global scientists started working on this problem. They want to know the structure of DNA because this is the molecule of life. The life came to planet Earth because of this DNA, right? So then I will shift this story to a different one. So there is a university in America, Oxford or Cambridge. In that university, just like you, there are some 2 PhD students. They are also working on, they want to know the new research, they want to do. Then all people started working on what is life. Already Darwin's evolutionary theory has been published, but still don't know what contributing to that life. That molecule no one knows. But already Darwin becomes celebrated. So things are very hot. Everyone is very serious about cracking this story. Then what happened? 2 students, they want to know what is the structure of DNA. Meanwhile, there is a woman. Her name was, she is the heroine of today, Rosalind Franklin. Rosalind Franklin. Who is she? Keep in mind, 100 years ago there is a huge gender discrimination. They saw that women are not fit for studying. Women are not fit for science. Women are someone low class. They are only for home, kitchen, babies, taking care of children. That is the time. In those times, Rosalind outshined in her academics. She is the only one who got PhD in Cambridge, and no one likes her because she is a woman. And you know what happened? She got her PhD degree in crystallization. She became expertized in X-ray crystallography. X-ray crystallography. What is this X-ray crystallography? Very, very, very interesting. So what they are doing was, the mobile recording and I was there. Torchlight. Anyway, let us assume we need to understand a shape, a structure of an unknown molecule. So we can do one thing, very simple thing. First, we'll keep in one direction that molecule and we'll focus a light over it. And let us observe the shadow. We are taking a photo of a shadow. When I keep book like this, I will capture a shadow. Maybe I will capture this shadow. Then I will rotate my object to 90 degrees now. Then I have taken one more photo. Now I want a photo like this. When combining this and this, now I came to know this is a structure of containing like this. So taking 2 photographs in 2 different angles, then we can able to construct its 3D structure. Simple idea. You got my point? If it has hyperstructure, we will take in multiple angles. The photos in multiple angles. We will capture the shadows. We will reconstruct the shadows based on the rotation. In what rotation we have tilted. So by piecing all this information together, we can understand the 3D structure of a molecule. This is called as X-ray crystallography. Here, instead of taking a light, we are taking an X-ray because X-rays has a very shorter wavelength, lesser than a light, visible light. So we can able to see the structure of minute atoms. It is possible to see the picture of a molecule. Got it? So then, our husband finished her PhD in X-ray crystallography. She is very good at X-ray crystallography. She is understanding physics actually, of coal, minerals, inorganic things. Then, her first job. She wanted to pursue her job in research, and she went to her first job. And there is a scientist called the guy name I forgot. Maurice Wilkinson's. So she joined in his lab. I want to be a researcher,
research fellow in your lab. And you know what Murray said? Okay, you have good marks, but you are a woman, I won't give you any scientific position. I will give you only a labor, a technician job. Are you interested? She was very sad. She wrote in her autobiography. The world was, very painful, the world was cruel. But she is very passionate about science. So she started working as a technician after doing PhD from Cambridge. 100 years ago. She outshined everyone, but keep in mind, how she got good marks. Everyone tried to give her less marks. Still, that is how hard she worked to get the degree. And what is the award she got? A technician job in the lab. But she is not so sad. She was a very strong lady, such a passionate lady she was. So one day, she started turning her head towards a leaf. She took a plant leaf and she wanted to know what is the structure of DNA. She took the cell, she took the nucleus, she kept it in a mold and she started bombarding it with the X-rays. Under the supervision of Wilkinson. Wilkinson will say everything because he is the manager of the lab. So what she started doing? She started taking the pictures of multi-dimensional photos of nucleus. And she wrote in her book that perhaps it might be a helix. She wrote it might be a helix because she got a photo of X. She got a shadow, X-like shadow. Only a helical structure will give a shadow X. So in her book she wrote, in her writing notes, it might be a helix. So she almost cracked the story of DNA. Okay? Because we already know everything. Just we need to know how they are organized. And because of her prolonged 5 to 6 years of X-ray exposure, at that time there is no research on cancer related X-ray related cancer. So they are unaware of X-ray related cancers, and she got a ovarian cancer actually. And she was admitted in the hospital, severe abdominal pain, and she was admitted for 6 months, where she kept her data in the lab and she went. Okay. Meanwhile, the two people from Cambridge University, they also want to know what is structure of DNA. So one day they came to this town where this Rosalind lab was there. When they came to this lab, this Marais, he said, my lab technician working on some data, she's not so smart, she's useless only. Maybe this data might be useful to you. See her data. So our James Watson saw the picture and he started smiling. There is an X shape and there is a helix. He started smiling. Next day they went back to their lab. Within 4 days they published an article, the structure of DNA. They cracked the structure of DNA. And within 6 months they got Nobel Prize. Interestingly, Nobel Prize will be given to 3 people for a single discovery. And you know what happened in the Nobel Prize? The Nobel Prize awarded to Watson, Crick, the 2 friends, and the third person who deserves here, the third person who deserves the Nobel Prize for DNA discovery. Rosalind, right? The Nobel Prize was given to Marais Wilkinson. And she passed away after 1 year after this. She is no more. Yeah, there is a huge betrayal in the science of DNA quest. And none of the textbooks, highlights Rosalind's contributions. There is a problem in the history. Just like DNA is twisted, the story of DNA also twisted. Because there is an involvement of women. See? People used to say that she is the dark lady in the quest of DNA story. But some people used to say she is the brightest lady. People label her as a dark lady rather than bright and shiny. Yeah, that's happened. Yes. Actually, there is a dedicated story for Rosalind. I will tell you in one good day. So, now they understood what is the structure of DNA. So, what James Watson said, James Watson and Crick, Watson and Crick model of DNA.
They say that DNA has helical like this. Okay. I don't know how to show it. There are two types of helix. One second. I really have this laptop. Listen. So, structure of DNA is like this. It is in helical shape. Yeah, it is in helical shape. Two layers. It has two phosphate layers. These are the phosphate bonds. Keep in mind, this is just a pictorial representation. Actually, DNA won't look like this. It's the ribose, sugar, phosphate, and base pair. Okay, but for pictorial representation, we are showing in this way. So this is the phosphate backbone. Phosphate backbone. Then sugar is already there. Sugar and the rest together. And this is the base pairs. ATGC base pairs. Now this has two types of groups. If we plot a straight line from here to top, if we plot a straight line, there is a major group and there is a minor group. So DNA has a minor group and a major group. This is called major group. Okay. Then it has a hydrogen bonds. Triple bond in between GC and double bond in between AT. And the minor group is 0.34 nanometers and that is 3.4 nanometers, and the diameter of DNA is around 20 nanometers. Something that you need to bifurcate. This is the basic structure of DNA. Clear? Any more doubts in the structure of DNA? So this is the actual structure of DNA. A segment of actual structure of DNA, right? So here you can clearly see a sugar base, a phospho-sugar backbone. This is the sugar link and this is the phosphate. So phosphate, sugar making a backbone. Then we have a base pair, one to this phosphate backbone and one for this phosphate backbone. So there are two types of base pairs. If it is A, this will be GCAT. Then, in case of RNA, the tyrosine is replaced by uracil. Tyrosine was replaced by uracil in RNA. You know, right? This one. Tyrosine is replaced by uracil in RNA, and all other things are same to same. Okay, this is the structure of DNA. Okay. See, that's it. Then next one, we need to see, when we count 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So there are 5 carbon rings, 5 carbon bonds. So based on this one, this position of the carbon, the two strands are divided into two types, 5 prime and 3 prime. One strand will go from 3 to 5 and the other one will be, if it is 3 here, it will be 5 to 3, 3 to 5. So anti-parallel strands. There are two anti-parallel, 3 prime to 5 prime and 5 prime to 3 prime, and phospho, sugar, base pairs with nucleosides that include ATGC following the Chargaff rule. Any doubts so far? Yeah. So let me open this ChatGPT notes. See, you cannot directly study from ChatGPT because it is a very basic course. First, you should understand the textbook. When you are learning a huge data, when you are unable to understand the data, then ask GPT. It might give in a simple form. But don't rely directly on the GPT answers because it's useless. It will give very basic knowledge only. Got my point? I'm not saying don't use it, but just use it as a tool to understand the topic. But it will give only basic information. You can't gain excessive knowledge from GPT. Yeah, that's what I want to convey. And let us see the notes now regarding structure of DNA, and let us compare with our class. So yeah, see here. The DNA, okay, DNA is a double helix, a highly organized, stable, essential structure for storing and transmitting genetic information. Simple. Discovered by Watson and Crick, damn, you know, where is Roswell fragment? That is very painful whenever I am saying history. It is composed of two antiparallel strands made up of nucleotides. Antiparallel strands made up of nucleotides, right? Each nucleotide consists of three components. Each type has three components. What are those? A phosphate backbone, a sugar, and a nucleoside. So they wrote here. I forgot to tell this thing. Just keep in mind. Adenosine and guanosine is called as purines. I didn't say this previously, sorry. So adenosine and guanosine together are called as purines, whereas cytosine, thymine, and uracil are called as pyrimidines. So AG are purines, ATR or CTU are pyrimidines. CTU are pyrimidines. Is it clear? This class is still